Thank you all for coming today. For those of you that don't know me, I'm Peter Vickesland, and I'm a professor of civil environmental engineering at Virginia Tech. And I just want to thank all of you that are both here in person and virtual for attending this workshop. Essentially, this is a workshop on human engineering and scientific aspects of disease transmission in natural and built environments. And this is a workshop that was put together on behest of NSF. They essentially wanted to bring kind of the environmental engineering and science community together to talk about disease transmission in both the natural and the built environments. So essentially, we're here because we know that, and we are very interested in how diseases are transmitted in both the indoor and the outdoor environments. We want to get a better understanding of those transmission processes. And we really want to figure out how to develop approaches to essentially address disease transmission. So we need to understand the science, the engineering, and kind of the human factors that are important in all of that. And we want to do so while thinking both ethically and sustainably in that effort. Now we know that diseases are transmitted in the natural and in the built environments. And those disease agents that spread essentially can be viral, they can be bacterial, they can be eukaryotic, and effectively, we want to understand those transmission processes across a variety of different routes. So I've illustrated up here transmission by water, transmission by food, transmission by air. All of those are different media by which these infectious agents can be spread. So one of the things that we know, and we've seen for the last few years, we've seen for literally decades, is that environmental engineers and scientists spend a lot of time thinking about how to quantify those processes. Now, we know, as I said on the previous slide, that disease agents spread outdoors, but they also spread indoors. And effectively, we are interested in trying to quantify those processes indoors in a variety of different ways. All of the red dots that are illustrated on this slide are trying to show effectively ways that disease agents can come into an indoor space. So they might come from outside, they might be present within the fungi that grow in the carpets or on other parts of the home. They might be aerosols that develop when you flush a toilet or you essentially take a shower. All of those are possible ways that we can think about disease agents getting into the indoor space and affecting people. And we can also, looking at that slide, just think about ways that we can try to mitigate some of that spread. Now we care about the indoor space because, as Rich Corsi, who is the current Dean of Engineering at UC Davis, has pointed out, in average, an American lives to be 79 years of age. And of those 79 years, people spend 70 of them indoors, 5 outdoors, and then 4 essentially in transit, going from place to place. Now, of those 70 years that we spend inside, 50 are spent at home, and then 26 of those years are spent in the bedroom. Most of that's going to be sleeping. But again, 89% of our time is spent inside. So we need to think about what happens in the indoor space. Next. Now, we also know that respirable infectious diseases are going to be spread by airborne transmission of disease agent-laden aerosols. And we're going to learn quite a bit about that today and tomorrow. But effectively, if we want to minimize the spread of such diseases, one of the things that we need to do is really think about understanding what happens outside of the body. So the medical community has spent a lot of time thinking about what happens inside of a person, but we also need to think about what happens outside as well. So, next. To do that, we really need to be thinking about doing things across multiple scales. And effectively, as we think about what happens in a suspended aerosol, we need to understand the chemistry, we need to understand the biology, And we need to understand the physical processes that are going to affect the viability of that infectious agent. What I've illustrated on here, I shouldn't say I illustrated, what has been illustrated on this slide is essentially an infective virus in an aerosol. And we need to think about the transport of that virus within the aerosol droplet. We need to think about what happens to that droplet as it is suspended in the air and then ultimately settles on the ground. We need to think about the chemical interactions that are occurring within that droplet, both in the interior as well as the exterior of it. And then we need to think about its physical interactions. So what physical processes affect the fate of that aerosol droplet? So this is very much at the molecular or very small scale. But as we go further out, next slide, we need to think broader. So essentially, if we are this infected student that is ill in this classroom, we need to think about all of the different processes by which those aerosols are going to spread within the indoor space. And this is just an illustration for suspended aerosols, but we can think about these same kinds of things in water, in food, and so on. But we really need to be thinking across a variety of different scales if we want to understand what's happening and if we want to develop control strategies. Next slide. And then last but not least, we actually need to think globally because we have people that move across the globe. We have commerce that's going to be international, things moving from place to place. And with that, we have the spread of infectious disease. So all of these things are happening. Global scale, regional scale, local building scale, and then way down at the scale of individual aerosol droplets. And that's what this community spends a lot of time trying to think about. So next slide. Now, if we're going to study all of these things, there's a lot of work that needs to be done to develop and deploy different types of tools. And essentially, a lot of the work that my lab has done over many years is to try to develop new ways to detect pathogenic organisms in a variety of different media. But we also have developed new tools in terms of PCR, new approaches in terms of metagenomics. And we're going to hear about some of those over the course of the next day and a half. But all of these new tools are great because they're allowing us to detect and look for things in places that we might not historically have done. And in fact, as we saw during the pandemic, there was a lot of effort placed towards developing approaches to detect SARS-CoV-2 RNA within wastewaters. And it essentially led to the protection of a lot of university communities, larger scale things within cities. And this is effectively developed into this community that's very interested in deploying surveillance techniques to look for COVID, to look for influenza, to look for other infectious agents within sewage. But as we'll see, in the future, we need to think about what that means. But one of the things that we can see based upon the work with COVID monitoring, based upon other things that have been happening, is that we can readily envision a future where we're going to have the capacity to detect disease agents in situ, in real time, or in near real time. And this was a schematic that I produced a number of years ago where it was still kind of in the far future. But now we're getting closer and closer to this every day. With that, we can expect that we're going to be able to prevent future disease outbreaks. And we can think about detecting coronavirus in indoor spaces like we're in today. There's now real-time approaches to do that. We know that we can use wastewater monitoring to detect a variety of different diseases. And there's lots of effort to deploy this to go and detect and prevent the spread of antimicrobial resistance, which is thought to be potentially the next pandemic that will affect all of us. A key challenge, though, as we're trying to develop and think about these approaches, is that we really need to think about their ethical, legal, and broader societal implications. And essentially, we'll hear later today about some of the ethical issues associated with wastewater-based monitoring. Further, though, we also really need to think about a lot of the challenges that we are going to face as we start to deploy sensors and detect things in a variety of these different matrices in terms of privacy. And so there was a paper that came out just earlier this year in Nature, Ecology and Evolution, talking about inadvertent human genomic bycatch, where essentially you can find human DNA in a lot of these samples, and you can go and see who's sick, where they've been, who was there. And essentially that's led to a lot of thoughts about what are the ethics of detecting these human sequences in these kinds of samples. And one of the author of this article will be speaking later this afternoon. So thanks. So we've organized our workshop around 4 themes, and these are highly interrelated, which makes sense given a lot of what I've just presented. But the first is the fundamentals of disease transmission in the indoor built environment. The second is engineering approaches to mitigate disease transmission. The third is the monitoring of disease agents or markers in environmental settings and media. And then the fourth is really trying to think about how human behavior and risk play a role in all of this. And so the speakers have been kind of organized around these themes. And I should point that I probably could have put almost any one of the speakers under any of the different themes because they all work in these different areas. But for the first theme, we'll have Kim Prather from UCSD give us a keynote this morning, C.Y. Wu from the University of Miami, and Karen Dannemiller from Ohio State. The second theme on thinking about mitigating disease transmission, Claudia Gunsch from Duke and Yun Shen from George Washington. And then in the third theme, the monitoring of disease agents or markers in environmental settings and media, Aaron Bivens from Louisiana State, Creston Mansfeld from University of Colorado, and then Amy Pruden from Virginia Tech. And then the third theme, or the fourth theme, I should say, the consideration of the importance of human behavior and risk, Carrie Hamilton from Arizona State, Natalie Ram, who is a lawyer at the University of Maryland, and Shalini Misra from Virginia Tech. And Shalini actually is more in the convergent side of things. So thinking about trying to bring all of these different themes together. So my charge to everybody that's here is really to try to think about and engage with one another as you think and talk about the human science and engineering aspects of disease transmission. To also think about how the themes overlap. As I said, any of the speakers probably could have spoken in any of the other ones. But think about how they overlap and how we can leverage progress in one to inform the others. And then I think the other part that I would hope that everybody does is to try to think about who's actually not here and whose insights are we missing that we want to have as be part of these conversations. And then the last thing, I just wanted to leave this quote, which was actually just in a opinion piece in Science that just this past week, essentially, effectively talking about the fact that scientists and engineers have not done a good job of describing the process of science and how essentially consensus can change over time. And then there's a little bit more in there, but it leaves off essentially that science advice requires dialogue, which is what we're all here for, and not data dumping. So with that, I will stop and I will turn it over to Mamadou.